Good morning, everybody. Hey, Alana. Okay. Let's get the chat up. Wherever that is. And let's share my screen. All right. Well, it's going to be a nice small group today. So everybody has to ask at least 5 questions. You all okay with that? Crickets. Okay, so depending on background noise, if you want, you're more than welcome to unmute your microphones just to ask questions as and when they come up. I don't really want anything kind of impeding your ability to ask questions. And because there's only 3 of you, I don't think we'll need to worry too much about, you know, confusing amounts of crosstalk and stuff like that. Lana and Michael, as you've just joined, where the hell's my chat box gone again? Dang it. More. Oh, there we go. So in that bar at the bottom, yeah, there you go. Michael's found it. There's the chat box, which if you want to ask questions in writing, right, rather than in person, you can do that as well. Okay, so first up, before we do anything else, let's get this back, get that in the way. All right, does anybody have any questions about any of the prior material, right? So on Tuesday, we went over genome structure and transposons. Does anybody have any questions about any of that stuff?
Okay, you can go first.
I had a question. It was on slide 4 of the eukaryotic genome structure.
Oops.
And it says, in eukaryotes, some components of the genome have base compositions which are usually high or low in GC content.
Mm-hmm.
What's the importance of that? Is it just because AT or more, like the big key? I don't know, like give more information, or why does it?
So it tends to reflect other stuff being there. So typically as an organism, you'd have certain GC content. I can't remember what humans is. I think it's like 45% or thereabouts. And if you find regions that deviate from that, almost certainly it's because there's a large amount of highly repeated sequence there.
Okay.
So those tandem repeats or satellite DNA doesn't always, but quite often is very AT rich.
Oh, okay.
Pardon me. So yeah, it's really kind of an insight into the history of that particular chromosome.
Oh, okay.
Essentially, you know, what went in there and so on and so forth. Also, yeah, I don't know off the top of my head if there's any correlation between coding regions and genes and GC content. I don't think so. But certainly there is between satellite DNA or microsatellites is their individual name. And they tend to be more AT rich. Don't know why, they just are.
Okay.
Alana, did you have a question?
Yeah, I had a question about the transposons.
Yep, where are we at? I
I
only just closed those bloody PowerPoint slides this morning.
I'm looking at PowerPoint 5.
Slide 5.
Yeah, or slide 5. I'm sorry.
That's right.
So
Okay.
I was trying to understand non-replicative transposition,
Uh-huh.
and I'm just wondering, when they move the transposon to another place,
Mhm.
what does it do to the DNA sequence that it leaves?
It leaves behind a tandem repeat. Well, actually, two flanking direct repeats. So let me go to slide 3. So this is why transposons can be so disruptive. It's not just the fact of having the transposon there, interfering with whatever was originally there. It's also when it leaves, it leaves behind those flanking direct repeats. So if you look at the bottom of slide 3,
Mhm.
where the speech bubble 4 is, so as a result of transposition in, you get these little overhangs, right, because of the staggered cut. So essentially the DNA is kind of cut like that, right? It's not cut
Mhm.
blunt like so, it's cut like so, and then the transposon goes in the middle. And so those single-stranded bits, those overhangs, get filled in, and that creates those flanking direct repeats. So you can imagine if you take out that, I'm kind of pointing at the screen, which is funny. There you go. If you take out that orange part, which is the transposon, and then stick it somewhere else, you leave behind those flanking direct repeats. And so that would add, in this case, an extra 5 bases to that piece of DNA. So if that's in a coding region, say an exon, then you have essentially 1 2/3 of a codon added to that reading frame. So it will cause a frame shift mutation if it's not a multiple of 3. If it is a multiple of 3, then it won't cause a frame shift, but it will add an extra amino acid, essentially. So usually it's not good if it's in a reading frame, if it's in an exon, like to code for protein. If it's in a regulatory region or just some random ass part of the chromosome, less of an effect, right? So if it's in a regulatory region, if it depends if it disrupts a binding site for a transcription factor or equivalent, which we'll be getting to in PowerPoint 15. So yeah, I mean it varies obviously, and the damage it does going in varies again in terms of context. Very much depends on where that transposon transposes as to the consequences of that transposition. That answer your question?
I believe so. So when it leaves or when it moves around, essentially the little yellow or orangey part on this slide is going to basically come out.
Correct.
Would it repair together with the flanking direct repeats seal themselves with DNA ligase?
Yeah, there's just the enzyme which cuts out, I believe, sticks the strands back together in like a blunt fashion like this. So as it's taken out, the transposase will just reseal that break. So it doesn't leave a damaged piece of DNA. It just leaves extra bases, and it's often called a footprint. Actually, if you scan a genome for those flanking direct repeats, you'll often find pairs of them kind of dotted around. And that's essentially where that transposon has moved around prior to when you actually look for it. So it's actually called a footprint. It's a mark that is specific to that transposon.
Alrighty,
Yeah.
thank you.
They're pretty funky things. That's for sure. Okay, any other questions on those two PowerPoints? Michael, did you have anything you wanted to ask about on those two? Or Rose or Alana, if you have other questions? You? Cool. Okay. Let's get those shut down. So free up some space. Okay, so these two PowerPoints are pretty chunky. You don't want this one. You can also learn about Xerofiles if you wish to. That's pretty fun. Where are we at? Here we go. Okay, so first question is, did you have a chance to look through these before this session? You can say no, I'm not bothered if you haven't had a chance to, but it will help me figure out what I need to do to help you learn the material.
I looked into chapter 15, but I haven't gotten to chapter 16 yet.
Okay, chapter as in the PowerPoint?
The PowerPoint, yeah.
Right, okay. How about you, Rose?
Yeah, it's the same thing. I like quickly glanced over the chapter 16 or the PowerPoint 16, but I didn't really like fully in depth read it. I only looked at chapter or PowerPoint 15.
Yeah, no worries. I only posted it up yesterday, I think. So I'm trying to get ahead to get all of these posted as soon as possible over the next couple of weeks, so you have everything that you need for the rest of the semester, and I can get caught up. So I'll just do this every time. I'll basically try and figure out what you've been working on, and then we can go through that in more detail. How about you, Michael? I know you've been struggling with getting connected and stuff. You there, Michael? No. Perhaps he's not able to. Let's see if he... Let's see. Well, maybe he had to go off and do something else. Okay, so let's look at 15 then. Okay, let's kind of reorient myself. But okay, so essentially this is all about the first half of gene expression in general, and the first half in specific, which is really transcription. Okay, so well, let's go with questions actually, right? Because I could just go through this again, but I've already done that with the audio. So what particular things don't make sense, right? What stuff doesn't have to be like specifics, but more general principles. What things did you find the hardest to get on your first pass through this? You can say anything. We can bounce back and forth. It doesn't have to be in order.
I was looking at slide number 12.
Okay.
And when in the audio recording, you went over all of the little bits and pieces, the introns, the exons. I understand all of the little pieces, but I'm having a hard time putting together exactly what's going on in this picture.
Okay. So this is often part of the problem that I have, because I forget that the bigger picture is not necessarily known by everybody, right? So essentially this is a picture of or cartoon really, of a eukaryotic gene. And so eukaryotic genes always go from 5 to 3 prime, always. It doesn't matter. So this is, let's go wind back a little bit, right? We've got double-stranded DNA. Can you see like the gestures I'm making on the video?
Yes.
Right, so it doesn't matter which strand it's on, only that it's going to say, if thumb end is 5 prime, it's always going to be 5 prime to 3 prime, right? So if it's on this strand, it's going that way. If it's on this strand, it's going that way. That's just kind of broader context, right? And we kind of get to that somewhere else in here. Can't remember where. Anyway, so a gene in general consists of two main blocks. One is the part that controls when it's turned on or not, which is the regulatory region, often called the promoter. The other part is the bit that codes for, well, mRNA and then protein, right? Does that make sense? So it's really information and how and when that information is used. So the first part, and it's really, really small here, promoters are actually enormous. In the dark blue, that's the regulatory part, right? We'll get to that later, particularly when we look at transcription in more detail, right? That's where the RNA polymerase binds. That's where the activators, enhancers, things like that. That's where those all bind. And my hands look huge. My webcam's like down at the bottom left. It makes it look kind of weird. Anyway, and then the rest of it, the blue, not the blue, the yellow, the green, and the red at the end, that's all the information part, right? Does that make sense so far?
So with this picture, is this kind of just like a blown up of multiple little sections within the DNA sequence at different points?
Yeah, so I mean, it's very much a cartoon because you don't have like these colored blocks in there.
Well, yeah.
Obviously, you know. So if you look at, sorry, I point with my pointer, this black line going through, right? That black line would be the DNA strand, right? And essentially, that DNA strand, obviously, you know this, it's made up of a sequence of bases, A, C's, T's, and G's, right?
Mm-hmm.
And particular parts of that sequence, and we often use this in reference to the start of transcription, which is +1. So, you know, 0 to minus, kind of like upstream of like 2000, let's say, is the promoter. That would be the blue part. And then the next piece in the DNA would be the 5' UTR. And then you'd find an exon, and then an intron, and then an exon, and so on. So they're all stretches of DNA that do different things, essentially, or code for different things. And
Okay.
so if you just looked at if you amplified up, say, 10 kilobases of DNA, right, out of one of your skin cells, in there you might find a gene, right, which stretches from one part all the way through almost to the other end of that piece of DNA. And so that gene consists of all of these different pieces or regions which do different things. Did that help with the context?
Yes, it did.
Okay, cool. And so when you go into that in more detail, you see those things do different jobs. And so you can then break it down to, okay, we've got the regulatory region, the promoter, that controls RNA polymerase binding and turning on and so on and so forth. You've then got the piece which encodes for the mRNA, right? And so if you look at the bottom, the line doesn't perfectly match up, but that green arrow right at the bottom, that would be your pre-mRNA, that would be your transcript, right? And we get into mRNA processing in PowerPoint 16 at the beginning of that one, right? So we won't worry about that too much right yet. Anyway, so that starts basically, if we had a little arrow on here, let's see, I might see if I can actually draw one. Now, look at this, in real time. Right, so that is Yes. Hope you feel special, Alana. It's just for you. Come on. Let's move that along a little bit, make some space. All right, can you see what I've done now?
Yes.
That like little worm arrow thing. Okay, that's the start of the mRNA transcript. So that's where RNA polymerase starts copying the DNA into mRNA, right? And it will stop kind of around here at the end of the 3' UTR, right? So that will be one entire eukaryotic transcript. You're right. It sounded like you fell over.
That wasn't me.
Oh, okay. I'll blame Rose. Rose. Anyway, so that transcript, right, is not in a fully functional state yet. And the reason is those introns are still present. So the exons are what code for protein. And you can imagine, right, if you just kind of do this as a thought exercise, let's say we started translation, which would begin at the beginning of exon 1.
Mm-hmm.
You translate that ribosome isn't going to know whether that bit of mRNA is supposed to code for a protein or not. It will just keep making protein based on the sequence of the mRNA. So if you want only the green to code for protein, you have to get rid of the introns. Right, so that's what it really means when it says that the eukaryote genes are interrupted, right? They're not one continuous coding region like prokaryotic genes are. So the actual protein coding information is only encoded by the green blocks of sequence labeled exon 1 through 4. The bits in yellow at the beginning and the end are made into mRNA, but they're not made into protein. They're used to help protein be made, right? They're part of the translation process, but they're not actually translated themselves. That's why they're called untranslated regions. Questions?
Hi, it's Rose again.
So
I had a
hang on a sec,
question.
Rose. Just let Alana, if she has any further questions about this.
Okay.
I think I got it through this picture here.
Cool.
Kind of.
Yeah, I mean, there's a lot going on in it. But if you remember the basic things that different bits do, it makes a lot more sense, right? So you got the promoter, you got the bit that makes the transcript, and you got the bit that makes the protein, essentially.
Alrighty, thank you.
Cool. Sorry about that, Rose. What was
That's
your
okay.
question?
I had a question, like, what is the reason behind, like, the 1+? I saw in the audio, or I heard in the audio recording, and in the PowerPoint, there was, like, a 1+ and, like, a -10 and -35 consensus sequences. Like, what is that all about? That's what I was.
Hang on. I can even do this on the PowerPoint. This is like real-time learning. There you go. It's the start of transcription. So that's the very first base that RNA polymerase makes into the transcript, into the mRNA transcript. So that's like it's like a starting point, you know, like the starting line of sorry, my brother's in Formula 1, a Formula 1 race, or the, you know, the gates of a horse race or something like that, right? That's where it starts. And so everything is referenced to that point. So if you have, you know, trying to get my hands in the right place, okay, transcript is this way, right? And it's all +1, you know, plus numbers. Anything prior to that in the regulator or in the regulatory region or the promoter, that's referenced in minus terms, right? Because that's number 1, then this is -1, -2, -3, -4, so on and so forth.
Oh, okay.
So it's really a way of referencing features in the DNA to a particular point, and that point is always going to be the same.
Oh, all right. Thank you.
Because otherwise it's really hard, right? Because you can't say it's in reference to this particular sequence, because that sequence will change from gene to gene.
All right. Thanks.
Yeah. Awesome. I'm improving PowerPoints in real time here. Let's save that so I don't forget. I'll upload that to Blackboard afterwards. Cool. Okay, what's next? Let's see what we got going on in here. Oh yeah, here's that diagram, Alana, that I was talking about in terms of directionality, right? How genes can be in different orientations, which is pretty neat. Okay, so another point, which is like the inverse essentially of eukaryote genes being interrupted. Prokaryote genes are not. They do not have introns. So when you get to PowerPoint 16, the bit of the start of that, which is about RNA processing, only applies to eukaryotes, right? Does not apply to prokaryotes, because prokaryotes have, you know, as the DNA equals mRNA equals protein, right? They're both, you know, they're all exactly lined up. And in fact, do we have a little bit? No, it's not in here. Let's just quickly jump over to this. Where are we at? These are someone else's PowerPoint, so I'm always a little unsure. Okay, so just ever so quickly, right, just to kind of expand on that, not only do prokaryote genes are collinear, so the DNA equals mRNA equals protein, but also they tend to be grouped together in what are called operons, so multiple coding regions kind of in a row, which is really interesting. It's essentially, it's a way of coordinating gene expression. So everything that's involved in one particular process, all of the proteins that are needed for that process are all in one single row, which will make one mRNA. And so if you want to, I think I used Trehalose metabolism, you could do fructose metabolism, let's say, or lactose or whatever. All of the proteins needed to do that, whatever that is, right? And they almost certainly need more than one protein. They're all encoded by genes in one big kind of cassette, essentially. It's called an operon. And so you make one single mRNA, and then you make all of the different proteins off of that one mRNA, which is pretty cool. Okay, any other questions? Questions? Rose, not trying to pick on you, but you mentioned about these -10 and -35 sequences.
Mm-hmm.
Does it make sense what those do?
Those were the bits of the DNA that the RNA polymerase recognized that help bind to the DNA? Oh.
Yeah, exactly. Sometimes it's RNA polymerase itself. Sometimes it's other proteins that help RNA polymerase bind. So in, where are we at? In bacteria, you've got two different things. You've got RNA polymerase and what's called the sigma factor, right? Can't remember which binds to which, but one of them binds to one consensus sequence and one of them binds to the other. In eukaryotes, it's called a Tadder-Box binding protein. So if you look at the top of this slide, you see something labeled TBP, that specifically recognizes that Tata box, that region there, which is at around -20 base pairs, give or take. And that binds first, and then that recruits the RNA polymerase and all the other stuff in to bind the DNA.
Oh, okay.
Yeah. So it varies a little bit between eukaryotes and prokaryotes, but the idea is it's what it's the minimum required to get RNA polymerase to bind.
Hmm.
That's
And,
why it's go on.
For the sequences, is it just like it has to have these sequences going like down, or like, for example, going down all the list of the genes that are there? It just is because all these genes have relatively similar to like the initial bases that are there, like the TTGACA
Right.
on the first gene is similar. Like, do all the sequences have to be the same, like in the same order, or is it just because they have relatively the same bases themselves?
Oh, okay. So essentially, if you do this for all prokaryotic genes, right, not just in E. coli and not just the ones listed here, pardon me, you will get those two consensus sequences pop out. So they're not going to have every single base in that consensus sequence. So if you look at the first one, which is LAC, involved in lactose metabolism, right? It's TATGTT, looking at the -10 sequence. But it will have most of them, and it will always be at the -10 region, right? So you know, give or take, you know, 1 or 2 base pairs. So it's always going to be at the -10, and it's almost always going to have, I don't know what the numbers are, like 4 out of 6 or something like that of the bases in that consensus sequence. And then you're going to have the same over at the -35 too. Now, obviously, there are going to be some changes. Those changes may depend on the actual enzyme, the protein that binds might be slightly different from one species to another. It may also be, I talked about, and also in here we go, promoter strength. Weak and strong promoters, right? So these are the two of the big things that control RNA polymerase binding, right? And so you have to have RNA polymerase bind for transcription to occur. And so you can imagine that, let's say, and this is kind of hypothetical because I'm really not an expert on prokaryotic gene expression, but let's say the closer to that consensus sequence you are, the stronger, more likely RNA polymerase is to bind strongly to that promoter, right? Okay. So you can imagine that, okay, this is the consensus sequence is what is required. And some genes are going to hew more closely to that than others. And that's going to be one of the things which affects how much gene expression comes out of that particular gene, right? Based on how readily RNA polymerase will bind to that promoter. So yeah, they're consensus sequences then, but they're not exact.
Oh, okay.
But they are shared amongst all prokaryotes, and they are always at those two positions. Right? The distance is really important because it's basically determined by the size of the RNA polymerase enzyme. Right? If you can imagine, RNA polymerase enzyme is going to be, if you can see like my cursor moving around, it's going to be like this sort of size or bigger. Right? So the distance has to be more or less the same for RNA polymerase to actually fit on it, you know, because it's a protein. Now we have something a little similar in, where are we at? Eukaryotes, but it's much more variable and it's smaller as well. And so that Tata box is both a recognition binding site for Tata box binding protein. It's also where the DNA helix is opened. Right, so if you notice both this one here in eukaryotes and the -10 sequence in prokaryotes are very AT rich. You know, it kind of makes sense really if you think about it. Same as origin of replication in DNA. You want a high AT concentration to make it easier to pull apart the DNA. Cool, cool beans. What's next? Oh, cool, Michael's back. Let's see if Michael wants to ask anything about anything else. If not, just keep them coming and we'll just go through them in turn. I'll point out one thing which, let's just get back to this. Yeah, termination transcription is a little tricky, but also transcription initiation in eukaryotes is a lot more complex. So as you can see in prokaryotes, it's pretty much a simple case of sigma factor and RNA polymerase binds. Maybe there's something else that promotes that. You know, you can get inhibitors and activators and things, but it's fairly simple. In eukaryotes, it's a lot more complex. It's actually crazily complex. Right, so Alana, you see what I'm talking about in terms of the promoter here? So in eukaryotes, you have what's called a core promoter. That's really involved almost exclusively in binding RNA polymerase, right? And the various associated transcription factors and stuff that help it do that. You then have this regulatory promoter that can go, in DNA terms, miles, right? That can be as far as potentially 1 million or more base pairs away, right? We're talking about, this is the about 20-30 base pairs difference here, right? So essentially that would be probably in my neighbor's kitchen, give or take, right? In terms of scale from my screen. So that regulatory promoter, that's where you get a lot of the fine tuning going on. You know, how much do you turn this gene on? Where, you know, what part of the body do you turn on? In response to what, right? So that regulatory promoter is really the key part in controlling the how, when, and how much of gene expression, right? And that's part of the complexity of eukaryotes, essentially, compared with bacteria. So in here, we've got a lot of different things. It's very much a sequential process. So you have the Tata box binding protein and one of the transcription factors bind. That then recruits this complex of proteins, which is RNA polymerase, a bunch of other transcription factors, and a protein called mediator, which you can see in that light purple here. Right. So once those are all bound, they are relatively stable on the DNA, but transcription does not start. So if nothing else happens, that basically hangs out there until it falls off, basically at some point in the future. What you need then are other proteins, and these are the proteins that bind to this regulatory promoter, right? They can be bind to enhancers, they can bind to response elements like bits of DNA that are recognized by particular proteins. like for genes involved in the heat shock response, for example, they would have a particular stretch of DNA that's recognized by transcription factors that turn on just those heat shock proteins. So once those have bound to DNA, or those particular stretches of DNA, that DNA is often then looped around, and then you have another protein bound to an enhancer. That essentially is almost like a, you know, like those old flintlock pistols. You know, that kind of comes round and it touches the mediator, and that's what starts transcription, right? So you can see how interfering with that by, you know, perhaps having negative regulators of transcription or the like, or a lot of activators, that's going to affect how readily that gene is expressed. So this slide here is a lot of stuff going on. It's very much a sequential process. It's in its simplest form, it's not too bad, but it's way more complex than if you look at prokaryotes in bacteria. All right, so that's one of the kind of nuggety bits, I guess, that you could talk about. And again, you can see that +1 here. That's the transcription start point. Let's just see if Michael ran anything back. Oh, cool. Anytime anyone does have questions, just pop up. That's really what I'm hoping to get out, or hoping for you to get out of these sessions, is being able to go over particular parts of the lecture and ask questions, much as you would do. Oh, hey, Simon. Much as you would be able to do in class, right? So the material's one thing, being able to actually kind of, you know, cogitate on and ask questions about that material is arguably more, well, yeah, maybe even more important, right? To get that clarification so you understand those things. Okay, next question. Questions. Give me questions.
I guess I have a question looking at slide 28. I'm trying to understand, it says that there's a string of A's in the DNA sequence that will produce the U's that will eventually terminate the whole thing.
Mm-hmm.
Is that string of A's just there, and when it reaches it, that's when it stops, or when does it know when to stop exactly?
The first one. So, and I don't actually really know why. Let's go on to 29. So essentially, if you look at the top, there are two features. So this is really intrinsic termination of transcription, right? It doesn't rely on any outside factors.
Yeah.
So we've got that string of A's here, right? And really it's kind of a little confusingly put that way. Really it's a string of T's, right? And those T's get turned into U's, right? Is probably a better way of thinking about it. I don't know why they would use the template strand, because that's just what's used to make a copy of the coding region. But anyway, that's neither here nor there. Typically, polymerases struggle with strings of the same base, right? And so when they get that, they tend to kind of pause or stutter. It's almost like they not quite sure where they're at, in a sense, right? So I don't know why T's or, you know, U's being produced are better for that. I really don't know. A particular property, perhaps. But in that particular gene, and you can imagine, right, this is the very end of that bacterial gene, right? We're going to have loads of stuff up here on the left, right, upstream, right? So this is a part of the sequence that is part of that bacterial gene, and it's just there, right? Kind of essentially waiting. Yeah, go away. So That's one part. We've got this string of T's, which is going to be made into U's, and that's going to pause the RNA polymerase. Now, we've also got another thing, which is these inverted repeats. Right, and so essentially what inverted repeats are, I'm trying to do it on screen, is they read the opposite in that way to that way. So that once they're produced, they come together and they form this loop, which you can see at the bottom. Right, this is often called a terminator loop. Right, and so essentially what happens is, I don't want to keep using my hands, but I don't really have kind of good space. I'll put my laptop a little bit further away so I have more hand space. Right, so the RNA polymerase kind of comes along, doing its business, and it produces a bit of mRNA coming out here, which contains that inverted repeat. So once, obviously, when the RNA polymerase is over that inverted repeat, nothing happens, right? But as soon as it gets past that inverted repeat and stops, that inverted repeat can form. It kind of goes zoop like that, and it kicks RNA polymerase off of the DNA. So those two things together allow this process to work, because if the inverted repeat wasn't there, the RNA polymerase would kind of stutter on the T's and the U's for a little bit and then keep going. Right. If the inverted repeats were there, but the T's and U's weren't, it would go past and that inverted repeat would form, but the RNA polymerase would have already moved on. Right. So it wouldn't affect the binding of the RNA polymerase to the DNA. Right. So it's those two things together. Pausing the RNA polymerase, and you'll see that's a function as well on row-dependent transcription termination. So we want to pause the RNA polymerase to allow whatever else is going to happen to do its job. And in this case, it's this formation of a loop structure in the mRNA that's being produced. Does that make sense?
I believe so.
Yeah.
The picture on 29,
Mm-hmm.
can you just go over it one more time? I'm kind of having a hard time where that loop is coming in at the bottom, but the majority of the U sequences are up here at the top.
Right. Yeah, I'm sorry. I want to have a pen. Actually, you know what I might do? I think I might figure out a way of getting. I've got a small whiteboard at work. I can strap it to the back of my bike. I'll bring it home tomorrow, and then I'll be able to actually draw this stuff out, which will be, I think, a lot more useful. So essentially, you need to. There's a bit missing in between in this image. Essentially, what you have at the top is the DNA strand which is being copied, right? The one that goes 5' to 3' left to right. Okay, that's my wife. And as RNA polymerase goes from left to right, it's going to copy that into a linear piece of mRNA, right? And so if I had a whiteboard, actually I need to make a note of this. Right, hang on. Give me just a second. I've got terrible, terrible memory. Yeah, I'm going to work tomorrow to look after my worms, so I'll be able to pick that up. Where are we at? Okay, back again. Hello, dog. This is my doggo. Where's she gone? Say hello, Luna.
Aww.
Yes. Super cute. She's like a teenager. She comes down at like, what is it, 10 o'clock in the morning? Yeah.
I feel it. My dog's still sleeping.
Yeah, it's like so lazy. Aren't you? You lazy dog. Anyway, she often likes sitting at the end of the couch, keeping me company, keeping my feet warm. Yeah, the whole household's waking up right now. I've got my daughter coming along too. Anyway, so getting back to this, what you need to imagine, what I would draw if I had the whiteboard, would be the mRNA transcript in between A and B, right? So that mRNA transcript is getting produced, and you can imagine it kind of being kind of, let's say, just at the bottom of that bit of DNA, right? And so going from left to right, you're going to get one part of the inverted repeat produced, and it's going to move along, and then you'll get the other part just as that RNA polymerase starts making those U's, and it pauses, right? And so that linear piece of DNA, as soon as those two parts of the inverted repeat are produced, automatically forms that loop shape, and in the process forces the RNA polymerase off, right? So the mRNA is produced in obviously a very linear fashion first, but oh, awesome. Thank you, love. My daughter just got me this. All right. This is a very interactive session. Okay, so let's see. Here we go. So essentially you have, there's your gene here. Right, I'll get my pens. No, is that back to front for you? Or is that?
I can see it, but you're still viewing the PowerPoint, so my picture of you is very small.
Okay, so let's stop the.
There it goes.
Stop the share. Is that better?
Yes.
Yeah.
Cool. Okay, so and I'll bring my many colored pens with me tomorrow as well. And so essentially what that's going to produce is this pen isn't very good, unfortunately. Right, that.
Okay.
So that would be the mRNA. I'm gonna have to press them really hard on this. Right, so that's the mRNA, right? But at the same time, obviously you have the RNA polymerase here, right? And as soon as that bit on that part of the inverted repeat is made, right? So it kind of, the RNA polymerase is going out that direction, right? Going that way. As soon as that passes over and has made that part of the loop, this forms this, this kind of structure like so, like that, right? So it forms this loop, and that loop forms actually inside the RNA polymerase a little bit. And because it's inside the RNA polymerase, it levers that RNA polymerase off of the DNA. It's like having a little screwdriver in there that kind of like just, or I don't know, you can think of lots of different examples of small things that force other things off. You know, essentially it forms inside that groove, and in the process pushes it off of the DNA. So that bit there, that's the missing part from that diagram, Alana.
Okay, that makes more sense.
Yeah. Cool.
And then I just have one more quick question. You say it doesn't like the string of A's or the string of U's, string of T's, whatever.
Yeah.
How would it respond to, say, a string of C's? Would it terminate as well, or would it just skip over it?
Well, I know for a fact that all polymerase, RNA and DNA polymerases, don't like repeats very much. They tend to stutter and pause and make mistakes, essentially. Why U's in particular? I don't know. It may be a particular property of a uracil base that makes that happen, makes that pause more likely, maybe. I'm just guessing here, to be honest. I don't know, but it is a pretty common feature of these termination. And actually, if you think about it in terms of evolution, you're not going to find those strings of T's elsewhere in genes, because they would produce the exact same problem. They would cause a pausing of the transcription. I don't know how long they have to be to cause that problem. Again, that's just stuff that I don't know.
Alrighty.
Good question, though. All right, let's get back onto the question. So essentially, row-dependent transcription termination is a similar kind of deal. And here we've got another loop forming. We don't have a series of U's causing the same problem, but now we have a loop which basically causes the RNA polymerase to form. Just give me a second. Hey, Soph, sweetheart, would you mind just keeping it down a little bit? Just because I'm trying to talk over that to my class, okay? All right, thanks, love. All right. Well, my daughter's just getting a little bit exercised about something. Yes, so with row-dependent transcription termination, it's a little bit different. Here we have a protein called Rho, which is like a helicase, basically. And essentially, it's a racing game, right? So Rho will bind to part of the transcript which doesn't produce other structures, right? That's why it's called unstructured RNA. What that really means is that it will just stay a single linear strand, right? It's not going to form any kind of loops or funky stuff, right? So Rho binds to that, and essentially Rho is kind of here and RNA polymerase is here, and they're in a race, right? So they're going to be moving at the same rate. RNA polymerase along the DNA and Rho along the RNA, right? Trying to catch up, but as long as they keep moving, as long as RNA polymerase keeps moving, Rho can't catch it, right? And so now what we have is a similar deal to on slide 29, where you have these inverted repeats. But instead of kicking the RNA polymerase off, they cause it to pause, right? And you can see that in the middle section here. So that loop, called a terminator sequence, forms. I don't know why the one in intrinsic termination causes it to fall off and the one in row dependent doesn't. I don't know, perhaps it's kind of where or how it forms or its shape or something. I'm not really sure. Anyway, what that does is it causes the RNA polymerase to pause, and then Rho can catch up. And as soon as Rho catches up, it essentially takes the DNA template and the RNA coming off, and it forces them apart so that they separate. And in the process, RNA polymerase will fall off as well. So it's the same kind of idea. It's about essentially slowing RNA polymerase down for something to happen, but the thing that happens is different between the two. Did that make sense?
I think it makes sense.
Cool. And again, these are the same general principles kind of all through it, basically. You know, there's hang on, we got a chat. Oh, no worries. Let's get that further down there. It's the same idea really with eukaryotes as well. Eukaryotes are an awful lot less well understood, right? They don't have these terminator sequences in their genes to stop transcription. It's a lot more poorly defined. And so for eukaryotes, it's a little bit different. What it is now is part of the RNA processing, right? And when you go through 16, you'll see, how this happens. So essentially, once that mRNA is it's being processed, part of that processing is that it gets cut, right? And that cleavage site is defined by a sequence of bases that an enzyme recognizes, right? A very specific enzyme will only cut mRNA at that one point. It's not going to cut it anywhere else, right? So what's called an endonuclease attaches, cuts that, and then that provides a free end, right, for another RNAs called RAT1 to bind to it, right? So this RAT1 isn't going to bind to an intact mRNA, right? Because it's going to, it just can't recognize it. It has to find an end, right? And there ideally aren't any ends around, right? Because they're all going to be protected. So once this cleavage occurs, it's going to find a 5' end, and it's going to basically digest its way down the mRNA until it catches up with the RNA polymerase. I don't know, has anybody read The Langoliers by Stephen King? I don't know, it's what I always think about. It's basically like time is like previous days are destroyed by these things called the Langoliers. They go around eating up the world as it was so that, it doesn't get cluttered up, I guess. Anyway, it's a cool book. So essentially, this is again another race between the RNA polymerase and this I'm not sure why they call it an endonuclease. I would have thought it was an exonuclease. I'd have to check to figure out whether that was right or not. But anyway, it digests that mRNA faster than the RNA polymerase can keep making it. And if you think about it, that's a good thing, because otherwise that RNA polymerase, there's no intrinsic termination of transcription in eukaryotes. That RNA polymerase will just keep going forever and ever and ever, right, until it gets to the end of the chromosome, which will be hugely wasteful. And so this RAT1 breaks it down faster than it's made, and eventually it catches up and kicks it off. So it's a little bit simpler in a sense, but something that like as a heads up for future classes, it really pays to understand this loop formation. It's really, really important in the control of prokaryotic gene expression. You'll see it come up a lot over and over and over again. So it's an important concept to understand, this ability of RNA to form structures, right, depending on the sequence of that RNA. And I think that's pretty much it. Does anyone have any questions on any of this stuff? Let's make sure nothing. Oh, hey, no problem, Zolana. We're pretty much wrapping up right now. If you do have any questions about this, and I really strongly recommend going over this stuff again after we get off. These chats and the video is going to be posted. Nothing that's a private chat, obviously, but anything that is of any help to other students, that will get posted on Blackboard kind of as soon as I've finished doing this. Yeah, so if you have any other questions, make a note of them, bring them to the next session, and we'll go through those. I don't have any time limits on these, so we can do this for as long as is necessary. Okay, anybody else have any questions, thoughts? No? Awesome. Okay, well, thanks very much, everybody. That was awesome. I shall have a great weekend, best as you can. Stay safe, and I'll see you next week.
Alrighty, have a great weekend.
You too. Catch you later, everybody.